So the goal of today's lecture is to reinforce some of the messages from last lecture in the history of SARS-CoV-2, but the main goal is to dig a lot deeper into molecular virology of SARS-CoV-2. And we'll see what this means in a second. Molecular virology fundamentally, in this case, just means viral life cycle, which proteins it has, what does it do, once it infects the cell. And this is just basic understanding, and we'll keep coming back to it throughout the rest of the course. But I'll begin by going back to a paper, actually several papers that I'm going to discuss today were published before SARS-CoV-2, where coronaviruses were relatively obscure infectious agents, and they were thrust into the limelight in 2003, 2004, the original SARS and then MERS again. But because neither of those pathogens became endemic in the human population, the interest waned and kind of dropped out of existence. Not existence, but faded from the public view. So here is an interesting paper that was published. This is a review paper that was published back in 2018. And the purpose of this paper was to try to understand the distribution of different coronaviruses in various host species, and whether or not, how often do they jump species, and what can we learn by comparative analysis. So what they did was they collected, at the time, the most diverse sample of coronaviruses that they could get their hands on. They focused on our DRP gene. You'll learn a little bit more about it later today. So RNA-dependent RNA polymerase gene. The reason you pick a gene like this is because it's shared among even evolutionarily distant viruses, so you can align them and you can build trees. If you look at some other genes, they're just too distant to do this. And they built this large evolutionary tree. You go over what it tells you, because it's sort of a complex picture that it contains a lot of information. It looks pretty, but it actually is somewhat difficult to read. So one of the things, there are a lot of trees involved in coronavirus analysis, so being able to read and interpret these trees is an important skill. So the first thing that I would like to point out is that this is an evolutionary tree that contains different types of coronaviruses. So these labels at the very deep branches near. So here, can you guys see my my pointer? All right, great.
Yes.
So near the center of the tree is the root. So this point here at the very middle of the tree, that's the hypothetical ancestor of all coronaviruses. So if you recall, we mentioned at the end of the previous lecture, this is probably very, very ancient. So going back millions or tens of millions of years. So there's this branch goes to gamma. It terminates. They haven't actually looked at gamma because it's kind of not, it doesn't have any viruses that infect humans. All the human viruses are in the alpha coronavirus and beta coronavirus lineage. Obviously, because this paper was published in 2018, it doesn't include SARS-CoV-2. It might have existed in some form or the other, but we didn't know anything about it. So that's, and as you go down through the tree from the root, sort of following down these branches, you're getting closer and closer to the present, and all these leaves here, which are very small, it's actually a fairly large tree, are individual sequence isolates. So what's at the leaf of this tree is a particular virus that's isolated from a specific host. So this tree contains viral sequences, and there are lots of them, but each one of them came from a particular animal host. The vast majority of them came from bats. So everything that you see that's colored, all these little pieces of the tree, there are parts of the viruses that came from different bat species, and or genera, and there's a color code here. It's not terribly important unless you're particularly interested in the etiology of a specific viral species, which bat species or which bat genus a particular virus came from. Just know that they're diverse, both biologically and geographically. So I don't have a picture to show the geographic spread, but they basically try to cover the entire world. A lot of these sequences are from Asia, because that's where a lot of sampling occurs and where a lot of the bat species live. But all these colorful pies are from different bat species. You'll see some numbers and branches like 100. This is basically our degree of confidence and how certain we are that this branch is real. You know, the higher the number, the better. They won't show you, you know, too much if they are not, you know, well supported. And then you can start looking at this tree and identify some patterns. So one of the patterns that is obvious is that there are a lot of sort of solid colored pies, right? So all of these colors, individual like this, that you can see that are labeled by numbers like 8, 6, 10, and 11, they're all sampled from the same bat host species. There's a question, how do you isolate a virus without knowing what you're looking for? It's a very good question. I'm actually going to address it in great detail next time, because I'm going to tell you about how you do sequencing and viral discovery, but let's defer it until next lecture. So there are techniques for doing this. They're basically genomic techniques for discovering viral species.
Are there any non-mammal coronaviruses?
The simple answer to this is that yes, because there's no reason to believe there aren't. It's not included in this tree. And, you know, but based on the, you know, paper that I mentioned last time, the viral discovery where they went into snakes, well, not necessarily snakes are just one of them. And, you know, if we look far enough, we'll probably find something that's closely related. So there's a bunch of labels here on the outside, like HKU8, HKU10. These are names of viral clades, so they're all considered to be related viruses. There's a question about what's the difference between alpha and beta coronaviruses. They're basically different viral gene genera, right? So they're just taxonomically different. It's basically, you can think of this as, you know, we're part of the great apes clade, you know, and you know, alpha coronaviruses would be to primates, maybe like, I don't know, not quite mice, but something fairly distant, right? So when alpha and beta coronaviruses diverged was probably millions of years ago. So this is an ancient evolutionary divergence. But they're still sufficiently similar that you can tell them apart. So a couple of, and the other thing you can pull out of this is that, so they have these rings on the outside, which tell you, you know, how homogeneous is a particular clade by geographic area, host species, and host genus. And if it's light gray, it means basically everything is from the same area or from the same genus. So you want to look at some of the interesting things like SARS-related coronaviruses, where there are a lot of different colors, which means that, for example, these viruses, even though they're closely related to one another, they infect different bat species, which means there's a potential for species-to-species transmission. And you can also look at the white clades here that are not colored. These are basically everything that's not from bats. All right, so basically what you can summarize by looking at this tree, and there's a lot of work that goes into building this tree, is something along those lines. The distinct putative coronavirus species, so COV is a common abbreviation for coronavirus, tend to be associated with different bat genera. So basically a specific type of coronavirus tends to infect one or genetically similar bats, right? So that would imply something called codivergence, which means viruses infected the progenitors of bats a long time ago, and the bats diversified, the virus is diversified with them. As a general rule, coronaviruses exhibit genus specificity rather than species specificity, which means that they're still able to jump from species to species if the species of bats are closely enough related. And if you look at some of the individual clades like SARS-related coronaviruses, there's a high frequency of cross-species transmission in the evolutionary history of coronavirus, and this is something that I mentioned last week and probably something that you learned by reading the Nature paper that was assigned. And as of 2018, there were few observed recent transmissions, most likely due to poor sampling. It is important to appreciate, now we're moving to a different review paper also from before SARS coronavirus, that SARS coronavirus 2 is far from being the first coronavirus to infect humans and become endemic. Endemic is simply a word that it's transmitted between humans, right? So it's sustained transmission in the human population. Prior to SARS-CoV-2, there were 6 different coronaviruses identified in humans. 4 of those are endemic strains. SARS-CoV-2, which is now pandemic, you know, on its way to be endemic, makes 5. 2 of these were discovered in the 1960s. You will see that they're abbreviated HCOV, and then the species name OC43 or 229E. HKU stands for Hong Kong University. Not entirely sure what the others stand for, but I'm sure that they all have some naming etiology. And you have two epidemic strains, SARS-CoV-1, which caused viral pneumonia outbreaks nearly 20 years ago, and MERS 2012, which had about 2,000 cases, but very high case fatality rate, up to 35% in hospital transmission clusters, which means human-to-human transmission. Sorry, and there I've labeled them. So you have beta, alpha, alpha beta, and two betas. And SARS-CoV-2 is a beta. So endemic coronavirus, I'm sure you've had it in your life, probably more than once, because it simply is, it causes the common cold. In fact, up to 20% of tested individuals with respiratory disease were positive for some form of coronavirus. And because you've all had a common cold, you know what the symptoms are. It's relatively mild respiratory tract illness. You know, the common saying is with a common cold, if you treat it, it's 7 days, and if you leave it alone, it's a week, right? So it's, you know, your standard assumption with common cold is you don't do anything about it. There have been cases documented for all coronaviruses, including these benign common cold viruses, to cause fulminant or severe disease. But all the cases documented were typically in infants or immunocompromised individuals. So, you know, people that have pre-existing conditions or so-called comorbidities can have a severe progression or even a fairly benign disease. Here's a simple view of the evolutionary history of these viruses. You will see that there's a lot of uncertainty about where they came from. You will see that 4 out of 6 are putatively originating in bats. 2 of them are in mice. All right, so there's a murine coronavirus, which is sort of a distinct lineage. There are some hypothesized intermediary hosts, you know, maybe, maybe not. So camels, cows, these are civets, and unknown. And you have 4 endemic coronaviruses, and then you have 2 zoonotic coronaviruses, right? So in that case, they didn't establish themselves in the human population. So if you want to look at how the origin of a particular virus is tracked, we can look, for example, at something fairly benign. We can look at HCOV 229E, which was first described in 1966 by basically studying cultures from student volunteers who had respiratory disease. I believe this was in Holland, but I'm not entirely sure. Interestingly, in 2007, highly similar strains to this coronavirus were accidentally discovered in alpaca during an animal fair. They didn't seem to have spread very far, and nobody could really track down, you know, how they got into alpaca, but it does imply transmission from host to host. And incidentally, while people were studying a different disease, MERS, in 2012, and were sampling human hosts, they identified a camel isolus related to 229E. So this relates to the question that Alex had in the quiz. You know, in 1966, you could tell very little about where this virus came from in terms of animal species, other than to know it's probably from bats, because where most of them come. If you wait long enough and sample other species, you might have better resolution. So these sequences from camels, for example, weren't available until 2012. So in order to study the origin of a particular virus, you want to identify all quote unquote related sequences, meaning you can, they're sufficiently genetically similar, you can analyze them, and then you build and interpret a tree. And this is a simple tree here, much, much smaller than the one I showed you in the first slide. In fact, you only have 3 different species now. You have humans, you have camels, and you have bats. And there's a special sequence here called an outgroup. So this is basically used to orient the tree, to basically place the root at the oldest, at the proper point in time. An outgroup basically means a different coronavirus. So something that's similar to HCOV 229, but is not an HCOV 229. So as the way to read this tree is that you basically start at the leaves and you go back to the top, and as lineages what's called coalesce, so they go back in time, they merge. Eventually all the human lineages, which are 1, 2, 3, 4, 5 here, will merge at this dot, which is labeled with an arrow and titled MRCA humans. So MRCA stands for most recent common ancestor. It's a hypothetical sequence. It might not even exist, but you can think of this as the progenitor of all HCOV 229E human isolates. So you can potentially think it as, to the first order approximation, it's the first case. Not necessarily, but first order approximation. As you go, and you can see the same thing for camels, right? So there's 1, 2, 3, 4 sequences for camels, and they merge at this node here. So this is the MRCA of humans. And as you go further in time, here's the most recent common ancestor of all human and camel isolates. You're not 100% sure what species it was in. You might hypothesize it was in camels, but not necessarily in camels. You don't have any direct evidence for this. And as you go further back in time, eventually you'll merge with sequences from bats. So the most parsimonious, or sort of the most simple explanation for this, is that there was bat at the root, then it transmitted sort of to bats, then it went into camels, and then camels to humans and so on. But it is not the only possible explanation. But the important thing to read these trees is that you don't go top to bottom. So you don't, look at the ordering of the sequence, but you have to go back in tree and see where the most recent common ancestor is. All right. You could also, if you want to understand the evolution of host-to-host transmission, you can look for something that would be called unusual evolutionary events. So a usual evolutionary event would be a mutation. So one position in the progenitor strain mutated to a different position, to a different nucleotide in the observed strain. But you sometimes have these unusual events where whole sections of the gene become deleted. So for example, here you have a cartoon representation of a partial, or actually this is a complete viral genome, but it's a cartoon. So you have ORF1A, ORF1B, spike, you have all the accessory genes here. This little part is zoomed in. So you see there's spike, ORF4, and some other regions in ORF8, and things in red are deleted in camels and humans, but not in bats. And then if you look specifically at ORF1, ORF8, which is right over here, then you can see a different pattern of deletions where there's nothing is deleted in bats. Then you have a little bit deleted in camels, and you have a similar section deleted in alpaca. And then in humans, you have all of these long deletions. And you can try to stage them through the evolutionary time in the order of deletions. So, in the African bats, there were none. Then this one appeared. Then 1 and 2 appeared. The 1, 2, and 3, 1, 2, 3, and 4. And this gives you another handle at the origin of the species. And if you have shared deletions, it's actually much more informative than shared mutations because they're rare, evolutionarily speaking, rare events. So they're much more diagnostic, and there are a lot fewer of them. So the other thing that we know by looking at previous outbreaks is that once the virus gets over from the animal host into humans, you would expect that it needs to change some of its proteins to be better suited to replicate in the human. And there was actually in SARS-CoV-1, there was a beautiful example of this happening. And you can almost see it by simply examining the sequences. So here at the top, you have a cartoon of the spike protein, which you'll probably see too many times throughout the course of this, the course of this semester. But this is a cartoon. We'll talk a little bit more, actually a lot more today about the domains, so you'll have a better sense of what they do. But this receptor binding domain in the middle is the part that determines the specificity of the virus, so which host receptor will it bind to. And it binds to ACE2, which again we'll talk a little bit more about today. And both us, humans, and civets have ACE2 receptors, but they're somewhat different. So presumably, if a virus is adapted to infecting bats or civets, it might do better by changing some positions once it got into humans. So here we have each of these is a partial amino acid sequence of the spike protein from one isolate. So one sequence is one isolate, came from one individual. So there are two groups of human sequences. There were two outbreaks of SARS. There was the original in 2003, and it was a secondary in 2004. So there are three sequences from the first, three sequences from the second. There's an initial comparative outgroup from civets in 2003 and civets in 2004. And if you eyeball this alignment, you can basically see that the vast majority of the positions, they're the same between humans and civets. So the assumption is nothing changed, except for these positions 360, where there's predominantly an S in civets and an S in humans, was a special additional spillover event. And then in humans 2003, it's an F. And there are two positions in 472 and 479, which are different between civets and humans. The hypothesis here might be that these particular changes might have made the virus more efficient at replicating in human cells, meaning it's better at binding the ACE2 receptor. And by the way, if you're wondering what a civet was, here's a picture of a civet for you. Kind of looks like a long-tailed cat, but it isn't. So in this particular paper that's cited in the bottom, they've actually done what's called a mutagenesis experiment, where they have they hypothesize that changes at these positions modify the phenotype of the virus, so make it more or less likely to bind ACE2. And there are a variety of procedures for measuring this. They all require some sort of in vitro cell culture experimentation. So they do not do it on live viruses. They do it on some laboratory system. We're not going to go into details today. It's not particularly important, but we will probably circle back to this when we'll look a little bit more into pathogenesis, drug design and vaccines. But fundamentally, what you do here is that this is your experiment setup. You try to measure the rate of binding of the S protein that has some mutations but not others. And what you do on the x-axis is you titrate more and more protein, right? So this is nanomolar concentration. So you start at concentration of 3, and then you go all the way to concentration of basically micromolar. So this is an order of magnitude. This is a logarithmic scale here. And what you see on the y-axis is some sort of a readout, where the higher fluorescent intensity means more binding. What it actually measured depends on the assay, and again, it's not terribly important. You're looking at this qualitatively. So what they do here is they start, so this SBJ01 strain is an actual isolate from a human host. You can see it here. It's the second strain in the alignment BJ01. So this is a human strain. It's supposed to be adapted to binding ACE2. And sure enough, as it has highest mean fluorescent intensity, over low ranges, and then, almost everything else, almost over the entire range, with the exception what they're comparing to is the virus that is the same as human, except it has an F changed to an S at position 360. So it's right here, it's this position. That doesn't do anything to binding. However, if you start changing to other positions, so if you engineer if you change a T to an S at position 487, which is these upper triangles, you see you reduce binding. This curve drops. If you engineer the other mutation, which is presumably N479K, it drops more dramatic. But if you engineer both, you basically abrogate binding completely. So this is a clean experimental setup to show that these two, assuming that it's set up correctly, it's measuring what you want it to measure, that these two amino acid changes made SARS-CoV-1 better at binding to human ACE2 receptors compared to civets. And this is sort of, a lot of these types of experiments have been and are being done on SARS-CoV-2 to try to understand what all of these proteins do, because as you learn a little later, we understand what some of them do, but not what all of them do. So this, in addition to give you some backstory about coronaviruses and the fact that even if you haven't been infected with SARS-CoV-2, odds are you had some form of endemic coronavirus as a common cold. By the time SARS-CoV-2 emerged, there had already been extensive studies in existing human coronaviruses. So, we're not starting from zero. Host reservoir sampling, like what I showed you here, was very beneficial to origin tracking, and the role of bats in adaptive changes has been well documented. So once SARS-CoV-2 entered the human population, we didn't have to guess at a lot of things. We could basically take what we knew about other coronaviruses and just translate it here. Similarly, because SARS-CoV-1 and MERS were both pretty dangerous looking outbreaks, there was a lot of research following it to try to understand structure and genomics and molecular biology. So there's a large body of existing work and tools for genomic experimental analyses. In 2020, in addition to what was available before, we now have high throughput genomic techniques and computational analytics, which put together might lead you to conclude, justifiably in my opinion, that scientifically speaking, we're as well prepared for this outbreak as one could have reasonably expected. To contrast this, you might harken back to the 1980s. Of course, this is all ancient past for you, and I was, barely a teenager back then. But when HIV entered the human population, and it was discovered in the 1980s and characterized, it was basically completely from scratch with a new pathogen that was very different from everything else that people have seen before, and without the benefit of having all these techniques. So the pace of discovery that we're seeing now for SARS-CoV-2 have in part been enabled by all this prior research. So it's science always builds on previous work, stand on the shoulder of giants and whatnot. All right, so the rest of the lecture, I'm going to spend talking to you about the molecular virology of SARS-CoV-2. And this is largely based on a really nice review paper in the Journal of Biological Chemistry, JBC, I believe there's a link at the bottom. So it is fairly recent because the field is moving so quickly. Not all of this is perfectly up to date. But a lot of the things I'm going to tell you about apply to all coronaviruses. So there's nothing special about SARS-CoV-2. And in particular, I'll walk you through, the viral life cycle, which, how does the virus enter into cells? How does it regulate gene expression? What does it have to do in order to replicate? How does it interact with the host? What does it do in terms of basic immune evasion? What protein and protein products exist, functions, etc. And this paper, the group that wrote this paper, is also has a nice YouTube video, which is linked here. They call it Coronaviruses 101. You're always welcome to go and watch it. It's going to tell you a slightly different story. So the question is, when comparing and contrasting SARS-CoV-2 with other related viruses, what do you think contributed more to its pathogenicity, the increased affinity of the receptor binding domain or the polybasic cleavage site and additional O-link glycans? It's hard to say. I mean, I think you can probably find papers that argue that any particular, all of these changes contribute. Based on what has happened with other pathogens, I think we need to wait for the dust to settle a little bit in order to make this determination. All of these are reasonable contributors. It is also entirely reasonable to believe that there are other contributors outside spike that we haven't looked at. And keep in mind, again, that, for example, in terms of case fatality rates, SARS-CoV-2 is very mild compared, say, to MERS. Because if you remember MERS, it was in the tens of percents. It was really, really deadly. So we don't know definitively yet, but all of the things that you mentioned. So O-link glycans, that's probably what I would say is kind of the weakest one, because O-link glycans are, this is just sort of a general feature of viral envelopes. They bind sugars to code themselves and protect themselves from immune response. So typically you would think of glycans in the context of immune evasion. But well, I'll tell you a little bit about polybasic cleavage site and what it might do. So here's a picture of the overall life cycle of a coronavirus, right? So this is not necessarily specific to SARS-CoV-2. This describes coronaviruses more broadly. And a lot of these cycles are also not unique to coronavirus. I mean, all the viruses basically have to go through the same life cycle. So first thing, they have to infect the cell. And this is, don't worry too much about reading this because we're going to go through all of the individual stages in their own. but they have to infect the cell, which means binding to something on the surface of the cell and migrating through the cell membrane into the cytoplasm. Once they make it into the cytoplasm, they have to make their proteins so they can go through the rest of the life cycle, which basically involves replication. All right, so this is the virus makes its proteins. Then it goes to something that is actually technically called convoluted membranes. That is a technical term. At which point it does transcription and RNA replication in various cycles, again, which we'll discuss in detail. Then once all of this is done, the entire purpose of a virus in this point is to just make more viruses. So it copied itself. Now it has to make a new variant, actually lots of these variants preferably, which have to be packaged, assembled, and eventually released back so it can infect new cells. And as you look at viral life cycles, they tend to be surprisingly complicated, even though these are fairly small machines, genomically speaking. We're talking about, tens of thousands of bases of genetic information on the order of 10 to 20 proteins. But nonetheless, they're able to accomplish this fairly complicated looking life cycle, all the while avoiding the defense mechanism of the cell and hijacking its cellular machinery. A sort of broader level cartoon of the general life cycle of a virus. And again, this is now specifically, this is deliberately very high level. It doesn't even mention the virus. So you have viral particle, attachment, penetration, uncoating. So now you have, the genomic payload of the virus. You have to do gene expression replication, assemble back into a virion and release. All viruses use some sort of a cellular receptor to get into the cell. It's going to be quite different between different viruses. So for example, depending on which cells the viruses infect, will modify what they bind to. For example, HIV-1 binds a CD4 receptor, which is on the surface of the immune cell, whereas influenza virus binds sialic acid receptor, which are basically sugar receptors. Other viruses bind different receptors. And just as a cartoon, even the entry itself can be fairly complicated. So viruses hijack general transport machinery. So clathrin-mediated endocytosis is a general mechanism for cells to absorb external molecules. Basically, you have to be able to, traffic through the cell membrane. And viruses are able to hijack this mechanism and get into the cell. That's one way of the infection. It's not necessarily what all of them do. So let's look at the SARS-CoV-2 viral particle. Again, there's nothing particularly unique about this virus. They will all kind of look very similar. So the core of this is the viral genome. Depending on the virus, it's going to come in different arrangements. So here it's single-stranded genome RNA. That's all one molecule. In influenza, for example, you have segmented RNA. In HIV, you have 2 copies of RNA and so on. But this is all viral genomic payload. It's surrounded by, in the case of SARS-CoV-2, a double-layered membrane, which is transmembrane proteins, envelope proteins, and all of the viruses are going to have some sort of a protein that's responsible to binding to the human counterpart receptor. So for SARS-CoV-2, that is the spike protein. The other important proteins that are called out here are all called structural proteins, in the sense that they participate in the structure of the virus, right? Non-structural proteins are not present in the virions. They're only packaged here and they're expressed during the life cycle. So you have envelope, nucleocapsid, hemagglutinin esterase, and membrane. And here's another cartoon of how these are arranged on the genome. So the structural proteins are shown in green, right? So there's only 4 of them really, which begs the obvious questions like, what do the other ones do? And hopefully you will see. Some of them we don't know what they do. So the SARS-CoV-2 viral particle is coated with a nucleocapsid protein, which is enclosed in a lipid bilayer membrane containing 3 membrane proteins, spike, membrane protein, and envelope proteins. These are all called structural proteins. And for all coronaviruses, the M protein is needed to incorporate required viral components into new virions. So it actually serves multiple functions. In addition to being a structural protein, it's also involved in viral particle assembly. The N protein works together with the M protein to direct genome packaging during virion assembly. And you will see this pattern recurring over and over again. Because viruses have so few proteins, a lot of them take on multiple functions. And again, if history is any indication, as we study any particular viral system more and more, we will discover additional functions that we hadn't appreciated these proteins carried out. And it's actually quite tricky to discover the functions that you may not know are important. You know, since I spent so much time studying HIV in the last 20 years, you know, for example, we discovered what, you know, one of the gene does 20 years after the discovery of the virus, because we appreciated, you know, we discovered a host defense mechanism, which we didn't know existed. And that viral factor was there to counteract that viral host defense mechanism tethering. So basically, you know, what it did in that particular case is prevent viral particles from detaching from the cell surface. So that would be made, but that would never be released, which basically means failed viral replication. The E protein is a transmembrane protein that forms an ion channel in the cell membrane and participates in viral assembly. But now let's focus on the spike protein. For a variety of reasons, that's a very important protein. You know, from the standpoint of human hosts, they're important for us because that's what all the vaccines, or nearly all the vaccines that are being trialed now, are targeting. So we want to make sure we understand what it does and we're able to prevent it from performing its function. So the spike protein is essential for viral entry. It also determines host specificity, tropism, which is a term to describe which types of cell a virus infects, and contributes to important viral phenotypes, which we'll see what some of them are. So this is the first step of the viral life cycle, where the virus binds and enters the cell via membrane fusion or endocytosis. So endocytosis was the hijacking of cellular transport mechanisms that I mentioned before. So you have cartoon receptors, you have plasma membrane, and you have cytoplasm on the inside. So the virus is outside, needs to get into the cytoplasm to replicate. How does it do that? So for SARS-CoV-2 viral entry, and again, this is not terribly unusual for viruses. There are usually a number of steps that have to happen. So first, the spike protein is actually, you can look at it as one genetic sequence, 1,200 amino acids, but structurally it's actually quite complicated. It actually has 2 domains, which are fused together in the beginning, but then they serve distinct functions, and they're actually separated as a part of the life cycle. So the term for this is cleavage. So they must be cleaved or pre-cleaved by proteases, which is a class of enzymes that cut up proteins to function. The spike protein is also trimeric, which means 3 of the unit molecules that are encoded for by the gene have to bind together to form a function, which is why you see this little triple head at the bottom. So it actually exists as a trimer. It's actually also very common for viruses. For example, HIV envelope also exists as a trimer. So here's a cartoon depiction of what entry looks like. It involves 2 essential human proteins. So ACE2, which is the receptor on the surface of the cell. If a cell doesn't have ACE2 expressed on this receptor, SARS-CoV-2 or SARS-CoV coronaviruses cannot infect that cell. And it also has a host protease called TMPRSS2. Its job is to actually perform this cleavage. So this is the first step where the virus hijacks a part of the cellular machinery to perform its cell entry. So in fact, right? So this is, and sorry, before we go on to the next slide, also, I would like to draw your attention to this cartoon, which you saw in the paper the actual review that highlights the 2 subunits and the receptor binding domain. There's also polybasic cleavage site. So this is, again, one of these. If you read it left to right, this makes one of the 3 units that go into a spike trimer. So S1 subunit contains the receptor binding domain, so its job is to bind to ACE2. The S2 subunit mediates membrane fusion and enables the virus to enter the target cell cytoplasm. In order for it to enter the cell, spike must undergo not 1, but 2 proteolytic cleaving events. So the first one is you have S1 and S2 need to be cut up, and that's done by this protease. So once this occurs, then you have structural changes in S2. So it changes its conformation. It basically refolds. Then you can have an S2 prime cleavage, which activates it, leading to fusion and viral particle integration. So as we go through this viral life cycle, you can think of any of these stages that must occur in order for the virus to successfully infect a cell and replicate as a potential target for therapy. And all of them at some point will be or have been considered. So there is, for example, a class of fusion inhibitors for HIV again, that are typically, that were originally designed as drugs to treat individuals that had resistance to everything else. But it is possible to interfere with one of these stages and prevent a successful fusion or conformational change. So this is, again, a complex process. So once the ACE2 receptor binding is accomplished, S proteins have to be cut up into individual pieces. And it's important that this is done by the host cell surface protease. It has to do 2 cuts. Cut S1 off from S2. So S1 has now done its job. It can go away. S2 prime, then a spike becomes activated and leads to membrane fusion. Sorry, there's a typo here. And just a question for later, how can we learn about ACE2 and TMPRSS2? Some coronaviruses, for example, murine coronavirus, which is a separate lineage or separate species of coronaviruses that infect mice or MERS, are pre-cleaved at the spike, at the S1, S2 site by the cellular protease furin during their synthesis in the producer cell. So they basically have to do one less thing once they infect the cell. They're already pre-cut during their synthesis in the producer cell. So that's basically when they're made, and the viral particle is made. And notice this is a separate protein called furin, which is something, again, that I'm sure you've read in the Nature paper. Again, it has nothing to do with coronaviruses. It's generic cellular protease that just does that for all types of protein cleavage. But this pre-cleavage primes them for entry upon upon receptor binding. Coronaviruses, including SARS-CoV-2, appears to actually be multifunctional in this respect. They can either use direct fusion, like shown previously, or if they might also be able to piggyback on endocytosis for entry. And a lot of this appears to be dependent on whether or not they were pre-cleaved and some other factors which are not fully elucidated. So research on how often this happens is ongoing. SARS-CoV-2 seems to have much higher binding affinity to ACE2 compared to SARS-CoV-1. As of the writing of this review, and I'm not 100% sure if this has changed, the actual mechanism for why this happens has not been fully elucidated, because as I'll show you in the next couple of slides, the structure of spike actually looks very similar between the two. One possibility is that there's a unique sold bridge between ACE2 and SARS-CoV-2 spike, which doesn't exist in SARS-1, and then polybasic cleavage site. One of the interesting observations about these polybasic cleavage sites is they're seen in SARS-CoV-2, but they're not unique. They have been, for example, frequently acquired during experimental evolution when you passage viruses in culture to try to evolve increased transmissibility or get them to infect other types of cells and other viruses. And it's evolutionarily fairly simple. I mean, you don't have to make that many changes to the virus. As you saw, the polybasic cleavage site is just a short motif in the spike protein. So SARS-CoV-2 spike in any other protein, in order to advance our ability to understand how it interacts with cellular receptors, and especially to design vaccines and understand antibody specificity, you need to have a good three-dimensional structure of this protein, because it allows you to do all kinds of modeling and understand interactions. You can basically just have two molecules that are bound, and you can see how they attach together. These are very complex molecular structures, so modeling them, even with modern computers, is still a challenge. And some of these things you cannot answer without experimental validation. But protein structure can be directly solved by experimental techniques. The old one is called X-ray crystallography. It's been around, since at least the 1980s. X-ray crystallography requires you to be able to crystallize the protein, which sometimes is very difficult. So this has actually been supplanted with a new technique called cryo-EM, which is a direct imaging technique that requires some clever algorithms to impute the structure of the molecule. And to show you what this looks like, here's a figure from a science paper that came out in March of 2020, and it shows you a structure of pre-fusion structure of spikes. So what does it look like when it's just, outside, when it's not fused to the cell membrane? It's color-coded, so here's a genomic organization of the protein. So you have these units that are color-coded, and they play various important functions. So the green one is receptor binding domain. You can see it sticks out of the protein you would expect it to do, so because it needs to be available to attach to another protein, so you wouldn't expect it to be very deep in the core, that I wouldn't be able to attach to anything. Here's the N-terminal domain. Here's the S1, S2 junction. So now these pieces are now S2. And you can look at this, from different angles. And depending on how good you are, three-dimensional, spatial orientation, this may be easier or harder for everybody to see. However, what you can do, and I'll show you, in a minute or two, you can actually look at three-dimensional structures using 3D viewers and just twirl the molecule around to see what it looks like. So structures, and this is this is pretty much a standard step in every viral characterization. You solve its structure, you try to understand what all the units do, and you try to compare it to existing structures. Especially since in this case we had structures from SARS-CoV-1, which were solved 15 or so years ago. One of the things that you will notice is that the structure between two viruses that are quite genetically different. If you remember looking at the alignments between SARS-CoV-1 and SARS-CoV-2, there are a lot of changes, even between the isolates and spike. But if you look at the two structure, which is, which would be panel A here. So the middle is 2019 nCoV spike with a receptor binding site up promoter right there. And next to it in gray is the same protein from SARS-CoV-1. And if you just eyeball them, they look very, very similar. I mean, you can see that, this green part here is somewhat less angled than this green part. There's some, bits of the blue part that bind in the opposite direction, but they're actually very, very similar. And this is, again, very, very common for protein evolution, because structures evolve very, very slowly. You can change a lot of the individual amino acids, but the overall structure of the protein is going to remain largely the same. And this also makes sense because the way protein structures, functions is primarily determined by its structure and how the structure changes in response to external stimuli or binding to other receptors. So if the job of this protein is to bind ACE2 and then to go through all these other conformational changes, all the coronaviruses are going to have largely the same configuration. And in panel C, the two are laid right on top of one another. So you can see, it's kind of hard to see, but you can, here they've taken individual pieces, so N-terminal domain, receptor binding domain, S2 subunit, and overlaid SARS-CoV-1 and SARS-CoV-2. And it's really hard to see what's different between them, which is kind of the point. They don't change that much. All right, so at this point, I'm going to take a short break. The question is, out of curiosity, do you use the program PyMol for protein modeling? I don't do protein modeling on my own, but there are, I don't know, 50 different programs to do molecular modeling. I'll just show you one. So the one that's, so let me pause screen sharing and switch over to a browser. Make sure I go to the right window. Here we go. All right. So I'm going to search for PDB. All right, I'm sorry, but why does Trump and Putin show up when you search for protein data bank? That's an interesting oddity of that particular search result. So the protein data bank is a centralized repository for sort of standardized formats that describe three-dimensional structures of various proteins. Unlike every database that has anything to do with genomics, structure, or medicine, they call out specifically their SARS or COVID-19 coronavirus resources. So there's a nice banner in the middle, so we can click on this. And there's a whole bunch of different structures. So we can look at so they have accession numbers, just like GenBank. There are a lot fewer protein structures, so the accession numbers are shorter. But if you see something like this, this is when you refer, say, to a structure 5R84, this is a unique way to reference this particular structure. So let's narrow down a little bit so we can, we only want to look at structures from SARS-CoV-2. Let's say we want to use the ones that were done with electron microscopy that are proteins. Oh, there's apparently a release date in 2024. Not sure what that means. All right, and here are some of the structures that are available. So you can see what this is. Full-length perfusion spike trimer. For example, let's look at something like this if you want. So when you do vaccine research or try to study antibody binding, you will often try to study the complex of two molecules, a particular antibody. So this BD23 Fab is an antibody. It's a molecule that's designed to counteract spike. So it's coupled with spike. So we can click on this. And you can look at structure. So it's going to load in your browser. You can mouse over the individual molecules and you can see, so this is color-coded by unit. All right, so as you mouse over, there's going to be a window at the bottom that tells you that this is, SARS-CoV-2 spike glycoprotein. The mouse is right now over at valine, position 511. And this green bit here is the antibody. So this is actually a different molecule, and they're bound together in the complex. So this is, and the antibodies will have two chains, the heavy chain and the light chain. So the canonical structure of the antibody is a Y. So there's a light chain, there's a heavy chain. We can rotate it to see better where it binds to SARS-CoV-2. It binds to SARS-CoV-2 right at the receptor binding domain, right? So you can kind of see, we're now in position 493, right? You can click on a specific residue. It is going to zoom in into very, very high detail, and you can see individual molecules and so on. So this is a powerful way to, interact with the viewer, right? And I personally find it, actually moving things around allows you to appreciate the structure, the proteins. You can also add, for example, if you want to, you can add molecular density, right? So this is what it would more accurately look like in terms of the space-filling structure. And when you do these types of structures, they're not 100% accurate. So they're typically going to have some resolution attached to it in terms of how precise they are. So root mean square deviation, but it's usually up to the point of a couple of angstroms. So it's, you know, atomic size. So these are very, very precise structures. And this is a great resource if you want to go and just understand visually what these proteins look like. And again, spike is a fairly complicated protein. If you look at, let's see if they have something that's a protease. So for example, here's a structure of the polymerase. So this is also going to be this is also be complicated because it actually involves multiple non-structural proteins. Let's see if we can find something simpler. And even these are complexes. There's got to be a, let me search for SARS protease. There we go. This is also a complex, now bound to a protease inhibitor. So this is a different viral protein that's now bound to a drug. Now we can also enable the density. So here's the drug and here's the protease. Different view, you can see individual molecules. Anyway, if you've never seen PDB, you can spend an entire semester-long course talking about structural biology, which obviously we're not going to do, but just be aware that this resource is there. And structural analysis like this is indispensable in drug design and to a large extent in vaccine design as well, because one of the things it enables, and we'll talk about it more later, because it allows you to understand which, how easy it might be for a virus to, for example, develop an escape mutation against a specific type of antibody. All right, I'm going to go back to presentation and let me check. I haven't checked the messages. Sorry, one moment. Too many windows. Sorry, I lost my message window and I don't remember where it is in the all right. Chat. There we go. Oh, that was a question. Little dots around the protein. That's just a way to display the molecular structure. These are usually some anchor atoms, or something that you don't display but just are aware that it's there. So the other thing that I'm going to mention and show you a little animation is that these structures are necessarily static because you freeze the protein in a particular configuration. For X-ray crystallography, you must do it because it has to be crystallized, so it can't be moving around. For cryo-EM, if it's wobbling around too much, cryo basically freeze it so it doesn't wobble too much. So you kind of take a still shot. So what's a movie of a protein? This is typically done by molecular dynamic simulations, which are very, very compute intensive. But they can basically make a movie of how protein will move and fold and whether or not they can bind specific compounds or drugs. All right, so I am going to go copy and paste a link, switch over to different screen share. So for example, and the link is right there so you can watch it yourself. So there's this company which runs is Anton 2 supercomputers, which you can see. So this is a state-of-the-art machine, but just to give you an idea of how much power it's needed to simulate something that's fairly fast. So there's a 24 1,000 atom system, it's just a large molecule, can generate 85 microseconds per day. And this is an actual hardware designed, it's just a purpose-built computer where the algorithms are actually built into silicon. So it's 180 times faster than a general purpose computer. So you run these simulations on purpose-built expensive machines, and outcome, let me see if I can find, there was a cool movie where you can actually see it unfold. So here's this one. You can see it wobble around and this part opens up. So this is a part of the fusion configuration. And notice there's a ticker at the top. So this is in microseconds. All right, so we're now up to 10 microseconds into the simulation. All right, so this takes significant amount of time. But one of the things you can do with simulations like this, and you can actually take them as a framework for studying how the protein configuration changes over time, right? And this is very important in order to, for example, model, binding or drug interactions and so on. So these molecular dynamics they're really cool, but they're very expensive computationally. And they're still just modeling. You can't really, directly measure anything. I'm gonna go back to the slideshow if I can. My screen sharing failed to start. One moment. There we go. Let me bring the chat back up. I need to work on my application switches. All right. So far I've talked about only the virus, but obviously it takes two to tango in this case. So you need to have human molecular components as well. And the most important one, basically step one, is the human host cell receptor, ACE2, which stands for angiotensin converting enzyme 2. So if you know medical terminology, angio usually means something that has to do with the heart. Tensin is increase or something like that. And ACE inhibitors are actually a class of cardiovascular drugs that are meant to lower your blood pressure. So to quote what this is, it's a cell surface peptidase that hydrolyzes angiotensin 2. It is expressed in most organs, with particularly high expression in the epithelia of lung and small intestine. So to translate into something more palatable, what ACE2 does, it lowers blood pressure by converting a peptide called angiotensin 2, which is vasoconstrictor, which means it shrinks the diameter of blood vessels, into angiotensin 1, which is a vasodilator, which makes blood vessels expand, right? If you tighten the blood vessels, harder for blood to flow, increase blood pressure. Dilate the vessels, drop the blood pressure. So because it's ACE2, there should be an ACE1, and there is an ACE1. So ACE1 and ACE2 do opposite things. So they're basically, positive and negative control. So ACE2 is a counterpart to angiotensin converting enzyme, which increases the amount of angiotensin 2. And ACE inhibitors are a class of cardiovascular drugs, for example, used to treat hypertension or arrhythmias or some other cardiac conditions. So one of the important things to notice here is where is this receptor expressed? Because, for example, some other viruses are only able to infect a very small subset of cells, the ones that express the right receptor. For example, again, HIV-1 is only able to infect CD4 positive T cells, which is a tiny subset of immune cells. It cannot infect anything else. Well, it can infect macrophages, but that's a different part of the immune cell as well. But it cannot infect, for example, non-immune cells. And here you have expressed in most organs, right? So it's potentially a promiscuous target for viruses to go for. And it's obviously something that you cannot, if you break ACE2, if you block ACE2, you're probably going to kill the host, because if you're not able to regulate your blood pressure, this is generally not a good thing. So I'll do one more screen switch, and after that, I will not be doing that for the rest of the day. And I would like to show you where to look up some information about where is my, there we go, browser. All right. So the first thing as I'm going to show you is Gene Expression Atlas. You know, if you remember the last 15 years, there was a large effort and a lot of money spent on genomic characterization of humans. And as a byproduct, we have very, very extensive databases of all kinds of genomic measurements from multiple species and multiple hosts. So in this particular case, it's hosted by EBI, so European Bioinformatics Institute. We can search for ACE2. And there's a lot of information about ACE2. So gene expression basically means which types of tissues can you find this gene being made, right? So this is done by expression analysis, so RNA-seq. Used to be done by microarrays, now it's done by a high throughput sequencing. We'll talk a little bit more about that next time. And it's used extensively in understanding you know, various organismal and biological processes. Gene regulation is fundamental. And you know, diagnostics and treatment. A lot of, for example, human diseases that have a genetic basis because of dysregulation. So there's too much of some gene being made or not enough of another gene being made. And there's also, interestingly enough, there are similar studies from other species, right? So you can look at cow, chicken, monkey, mouse, of course. So let's filter this down to see, for example, where is there a high level of expression? And you can see it's liver, lung, pancreas, rectum, skeletal element, spleen, and stomach. So let's see. And this is a color scheme over here that's done based on different studies. So each one of these is a specific study. You can look at the details. So there's an epigenomic roadmap, human protein atlas, human proteome map. And the brighter the color is, the more of ACE2 is expressed in a particular cell. Now, this study doesn't look terribly differentiated. It says that ACE2 is expressed everywhere else, but if there's variation, you look for dark, bright colors, and you can see you know, which tissues it's expressed in. Right, so that's one way to look things up. The other very useful resource is OMIM, which is a comprehensive authoritative compendium of human genes and genetic phenotypes. This is maintained by the NIH. You can type in ACE2 here. And you're going to get a very detailed report on ACE2. So you'll have, but this is from the human host perspective. So what does the gene structure look like? What does it do? What are the biochemical features? What's the molecular genetics? You know, animal model studies, and so on. So if you want to look up anything about what a particular human gene does, this is the go-to place. It is sort of a curated and maintained and very up-to-date resource of you know, peer-reviewed and supported information about all human genes and human diseases. All right, I'll switch back to presentation. The question is, do you think the ability of COVID to infect cells with ACE2 receptors is the primary cause of the presence of cardiovascular illness in so many COVID patients? Well, we'll talk about this, the various complications of COVID in a specific lecture where we'll just look at the human component. So let's defer all of that. There's a lot of somewhat conflicting information about the cardiac complications of SARS-CoV-2. So we need to address this extensively and carefully, but we'll discuss it in great detail in a specific lecture on pathogenesis. All right, so we've discussed step one. We'll go faster through the other two because step one is typically what you want to block, right? Once you've permitted the virus to infect the cell, things become a little bit more complicated. So successful vaccine will prevent the entire cycle from starting. It'll basically neutralize the virus before it is able to infect the cell. So the next step's like you're in now, okay? So what does the virus have? The virus has structural proteins. It basically gets rid of its coat, and now it has to build itself. It has to use the human cellular machinery to first make its proteins, which for coronaviruses turns out to be a very complicated process. Right, so this is before anything else can happen. So now you infected the cell, you basically penetrated the enemy line. Now you have to build a beachhead, if you're thinking in military terms. So set everything up, stage, and provision yourself so you can assault the rest of the enemy positions. So as a reminder, coronaviruses' genomes are long, so on average 2 times longer than most RNA viruses, so about 30 kilobases. They make, depending on the coronavirus, between 20 and 30 proteins. And they immediately run into a roadblock, which they can't simply hijack the whole cellular machinery to do everything for them, because they must make those individual 20 or 30 proteins. They can't replicate that on their own. They don't. Ribosomes are very complicated machineries that the viruses simply cannot afford to encode. So you have to use what the cell has. But host cellular machinery and ribosomes makes one protein from one messenger RNA, which means that if that's all you do, you're going to have one giant polypeptide that reads the entire length of the viral genome, and you're basically dead on arrival because that doesn't work. You have a bunch of proteins fused together. So the viral genome is only one ribosomal entry site. So the question is, how do you make multiple proteins? SARS-CoV-2 uses 2 mechanisms, one of which is fairly common. So you have fused proteins, large open reading frames, which get synthesized at once into a series of fused proteins, which are then later processed into individual proteins with proteases. And SARS-CoV-2 also has another mechanism called subgenomic mRNA production, which is very unusual for viruses. And it's almost unique to viruses as far as I know. None of this is unique to SARS-CoV-2, but is rather a feature of coronavirus in general. Again, this is going to be my constant refrain throughout this course, which is SARS-CoV-2 is not that different from existing coronaviruses in many of its features, other than, of course, to what it's done to the human society. But from a molecular perspective, it is not terribly different from SARS-CoV-1, for instance, at least at this level. So here's a view, again, of the viral genome. If you remember, the first half of it, or 2/3 of it, but from, about to position 20,000, is occupied by all the non-structural proteins, which come in the form of a very long reading frame. So non-structural proteins, which are abbreviated as NSPs, not very creative, I know, but that's how it's done. And the reason it's done is because when you characterize a new viral genome, typically you will find proteins by computational or some other analysis, and then you'll assign them a name before you know what they do. And then once you characterize what they do, you can name them additionally, but in this case, it's just the order of appearance. So non-structural proteins are encoded in ORF1 and ORF1A and ORF1AB. They are translated from the 5' end, the left, and can generate one of 2 long polyproteins, polyprotein 1A which is this segment here, non-structural proteins 1 through 11, or polyprotein 1b, sorry, 1ab, my typing has really suffered. The figure is correct, which encompasses all the proteins, all the non-structural proteins. Which one is produced is determined by behavior of the slippage site, which we discussed last time, which means about 20 to 50% of the time, depending on the coronavirus, you're going to have a -1 programmed frame shift, which will allow everything to read through to the end of the polypeptide and make everything else. This balance is actually not random, but it is tuned to maintain proper stoichiometry or balance between different proteins. If you look at this, every time you read through PP1, PP2, you're going to always make 1 through 11, and sometimes make 1 through 11 and 12 through 16. So it would be reasonable to assume that NSPs 1 through 11 are involved in more functions, and you need to make more of them than you do of the other polypeptides. And again, everything in the viral life cycle can be targeted potentially for treatment. I'm just going to mention it today in passing, and then we'll come back and discuss individual treatment strategies probably in 2 separate lectures. So drugs that target the frame shifting RNA structures have been explored, and it would be a general and potentially quite specific mechanism to mess up the life cycle of SARS-CoV-2. For example, if you're able to completely stop this frame shifting, the virus can never make these 5 proteins, and NSP12 is RNA-dependent RNA polymerase, which means if it doesn't make that, it can't copy itself, so it's done. For example, it would be very, very effective treatment if it were successful, for instance. So once you've made these proteins, now they're all stuck together, you need to block, you basically need to cut them up into individual pieces. And this is done by proteases. Remember, we already encountered one protease that does something on the surface of the cell to enable viral entry. These are now viral proteases, so these are encoded as a part of the payload. And SARS-CoV-2 and other coronaviruses actually have 2 proteases, which is somewhat unusual for viruses. Most of them can get away with 1. So you have NSP3, called PLPro. So it cleaves everything upstream of NS5. So it cleaves all of these guys. And this is the main, I'm wrong. Upstream of NSP5 means it cleaves everything from 1 to 5. And it's also involved in immune evasion. And NSP5 is a different type of chymotrypsin, so 3CL proteases, which cleave from 4 downstream. So this is the main protease. And protease inhibitors are a very common class of antiviral drugs. In fact, there's a number of various antivirals being trialed and developed against SARS-CoV-2 that are protease inhibitors. Very successful protease inhibitors have originally been developed in the context of combination therapy for HIV. So the introduction of protease inhibitors, I believe in 1996, is what basically made HIV into a manageable chronic disease, because prior to that, monotherapy was not viable. You needed to treat HIV positive individuals with multiple drugs, because otherwise the virus would simply develop drug resistance mutations in a couple of weeks. Now that we've gone through, we made some proteins, right? We split them up. Now these proteins have themselves to assemble into complexes and start copying the viral RNA to make new copies of the virus. So we're moving on to that step of the viral life cycle. And again, in this at this stage of the viral life cycle, coronaviruses are quite complicated compared to the other RNA viruses. So all of these NSPs, which now get their own names, some of them do. So for example, NSP13 is a helicase, NSP12 is RDRP, which is RNA-dependent RNA polymerase. This is methyltransferase. This is exonuclease. These things don't have proper names. So they all form a complex. It's not fully characterized yet. It's actually being, if you go back to PDB structures, there were some structures of these guys together that you try to understand how they complex together to actually make new RNA. So the complex of these non-structural proteins is called RTC, which is an abbreviation for replication and transcription complexes. They themselves, all this synthesis and replication occurs in a spatially sequestered structures called convoluted membrane structures. Yes, it is an actual term, that are derived from rough endoplasmic reticulum, ER, anchored in place by NSP3 and NSP4 and NSP6. Like other positive sense single-stranded RNA viruses, coronaviruses have to first make a complementary negative sense strand RNA, which is then used as a template to create positive sense genome to package in viral particles. So the positive sense genome is what you want to release. The negative sense is made as an intermediate stage and then it's degraded. According to this review, mechanism is currently not fully understood how this happens. And there's also parts of the viral genome, conserved RNA. So this is not a protein coding part of the gene. These are parts of the actual RNA of the virus. Structural features, including something that lives at the very end and at the very beginning and the very tail of the genome in the so-called untranslated regions, which are critical for replication and transcription. Again, it's fairly common to viruses, RNA viruses, to have these RNA elements in untranslated regions. And the end protein is involved in negative strand synthesis. Now here's a cool, a really sort of unique feature of coronaviruses, which is another mechanism to regulate things. So RTCs also synthesize subgenomic messenger RNAs, abbreviated to as SGM-RNAs, that encode open reading frames located in the tail end of the genome, proximal third of the genome. And here's a nice cartoon which I took from Wikipedia because they usually have quite good illustrations. This is basically what it does. So subgenomic mRNA means I want to be able to take this long template strand, which potentially contains multiple genes, and I want to be able to synthesize only parts of them, with the added restriction that the tail end on both pieces is the same. So this poly A tail and this 5' tail have to be identical. So you can think of this as nested primers for PCR amplification. So your subgenomic DNA mRNA means basically the ends are the same, but you're able to selectively skip pieces in the middle. You can think of this as a regulatory mechanism for viruses, which we as eukaryotes have in spades. There's a lot of regulatory mechanism that allows you to take pieces of mRNA and express them or splice them together. Good question. How does the cell know how to degrade the negative sense RNA? It's not entirely clear at the moment, again, according to this review. But I mean, RNA degradation is very, very common. So there are a lot of mechanisms that could do it. It's just not entirely clear at this point which one does it for the negative single strand RNA in this case. So you have RNA, as RNA-dependent RNA polymerase moves across the original template strand, it will occasionally posit specific sites called transcription regulatory sequences, or TRSs. You have two of them. There's one called TRS-L, which is at the 5' end. So this is the sort of anchored TRS. And you have TRS-Bs, which are interspersed through the genome, just near the 5' end of each ORF. What this allows to happen is that when our DRP comes across this TSR, this site and stutters, gives it an opportunity to do a template switch. So it can actually jump over all the way across to the beginning of the gene and shortcut the transcription. So here's another cartoon to view this. So you have, you're transcribing the genome, in this case from right to left. The parts 1 and 3 are always transcribed, and the variable transcription length occurs in this part 2. So you have RDRP complex chugging along. Here it comes across this TRSB site. It stutters, and in some fraction of cases will actually detach and slide all the way over to the tail of the molecule and drag this subgenomic RNA with it, and basically continues synthesizing from this point on. So this occurs in the negative strand. Then you start doing positive strand, synthesis from the 5' end, and depending on where this jump happened, you will have variable length subgenomic RNAs, which results in different levels of expression for multiple genes. So you might imagine it's not shown in this picture, but if region 2, you have, say, 5 different genes, depending on where you cut, you'll always make the one that's on the right. It will sometimes make this one, sometimes make this one, even more rarely make this one. So this is a way to do gene regulation and stoichiometry and dosage.
Okay, yo.
Sorry, if somebody has their microphone on, if you don't mind, turning it off so no one get background noise. I don't want to have to mute everyone. So this RTC complex is unusually complex for a virus. It's more similar to DNA-based replisomes than viral RDRP, which typically does most of the work on its own with just a small number of cofactors. So in coronaviruses, NSP12 is this RDRP. You can put it in cell culture and try to see how good it is at synthesizing RNA. It has minimal activity on its own, but if you add NSP7 and NSP8, it's greatly enhanced. And there are now, so you know that these two cofactors are necessary. And there's also now a protein structure of this complex that shows you that these guys bind together and form a replication complex. Much about the structure of function is still actively researched. These molecular mechanisms are notoriously difficult to study. They're difficult to model. And in order to study them in cell culture, you have to design very elaborate experiments. And the complexity of the experiment will increase with the complexity of the molecular structure. We do know that NSP13, which is a helicase, or you can think of this as an unzipping enzyme, it's shown here as unzipping double-stranded DNA, is another key component with not an entirely determined function. So the canonical function for helicase is to basically move in front of the complex and unwind the double-stranded structure. So the replicase can actually, bind to something and make a copy. So helicases are essential for this, to basically unzipping double-stranded DNA. Although there's some, because you can also have secondary RNA structure, like loops, that might complicate things. There's a potential additional function for helicase to basically navigate the complex around these local structures or obstacles. The specific roles of some other non-structural protein factors like NSP9 are still being actively investigated. And I'll pause here just to mention that, eventually we're going to learn everything about SARS-CoV-2 that we can learn with modern molecular techniques because everybody's doing research on it. But if you look at a typical viral system, a lot of the time, the one that's not a pandemic threat and the one that's not causing, trillions of dollars in damage and killing lots of people will usually not be studied to that level of detail. Pretty much we have, prior to SARS-CoV-2, the only viral systems that have been studied incredibly extensively would be HIV, influenza A, and to a lesser degree, influenza B and hepatitis C. Most of the other viruses are only partially characterized. So if you start looking at, what's in GenBank and what's another annotation, you will see a large number of viruses with genes of unknown function or hypothesized function. Another interesting feature of coronaviruses is that they actually encode a separate peptide, NSP14, that corrects replication errors. And one of the hypotheses for why coronaviruses have to do this is because their RNA is so unusually long, and they make these long stretches of continuously synthesized RNAs. An RDRP will make regular errors. I mean, it'll misincorporate a nucleotide, say, one every couple of thousand positions. So every time it replicates a virus, it's going to make a couple of errors, and that's just too much. It makes too many errors for the virus to be viable. Coronaviruses encode an error correction mechanism through this exonuclease. How do we know that it corrects errors? Well, first, we can just observe that the mutational rate in coronavirus is about 10 times lower than in other RNA viruses. So something is different about the way it does replication. So this error correction, by the way, isn't great. We encode a lot more DNA repair machinery, and this is essential because we live for many decades, and there are trillions upon trillions cycles of replication. So errors are made, but they're not made at this level of they're say 3 or 4 orders of magnitude lower when humans or other eukaryotes replicate DNA than when viruses do. So NSP14 performs error correction, and one of the ways you can test this is you basically take a laboratory system where you're able to measure mutation rate by just basically passaging viruses, and then you engineer a loss of function mutation into NSP14, right? So you break it, you insert a stop codon, or you simply cut it out. And then by breaking it in the murine coronavirus system in SARS-CoV-1, observably increase mutation rates. So this is how you would confirm it experimentally, right? You hypothesize that NSP14 has a particular function, then you set up a laboratory experiment in which you destroy this function, and then you observe that the phenotype is changed. And it is also a promising therapeutic target, because what you could do is if you're able to basically mess with the function of NSP14, means that the virus is no longer able to repair the errors, and that would basically be like giving the virus cancer, in sort of relatable terms, because now it has all these mutagenized viral RNA molecules that result in non-functional viral particles, and it basically dies. So I guess cancer-like disease is not a great analogy, because cancer-like disease is runaway growth. In this case, it's just mutagenic meltdown. So for example, you can imagine having an attenuated vaccine approach where you basically engineer a vaccine strain that has a broken NSP14. So it has presumably similar immunogenic properties as the fully functional virus, but it's not able to cause disease. There's some other tedious things that the virus has to do. For example, once you've made the mRNA, you have to cap it at the 5' end for stability. Because coronaviruses replicate in the cytoplasm and not in the cellular nucleus, where capping is basically automatic, they have to encode their own factors for RNA processing to make sure that it's capped and stable, it doesn't get degraded. So NSP 7, 12, 13, and 16 are thought to be involved in this process. So moving on, so this we talked about, stage 4, stage 5. I mentioned before that, and this illustration shows the double membrane vesicle, which are created in convoluted membranes. So what coronavirus does, it basically creates these bubbles with 2 membranes. It's not unique to coronavirus. There's other positive sense single-strand RNA viruses do this. It serves a couple of functions. It concentrates the replication machinery, so you can have more efficient replication. It hides double-stranded RNA from immune recognition. And it also spatially separates replication from assembly, because you don't want things to be, accidentally mixed up. You don't want partial products, for example, to make it into the assembled viral particles. And NSP 3, 4, and 6 are known to be involved in the formation and maintenance of this vesicle. And it happens usually just outside the nucleus. Once this is done, you still have to assemble a viral particle. So in this sense, SARS and coronaviruses are not as unique in some of the other mechanisms. So they use a fairly standard viral particle assembly, which happens in endoplasmic reticulum, or Golgi intermediate compartment complex, where you basically have a bubble formed. It gets studded within the structural proteins. Then you have a full viral RNA that gets delivered to it. It's tagged by the N protein, gets incorporated in there, assembled and released. What I would like to point out here is that even here you need to encode a special messaging protocol because at these life cycles that are not shown here, there's a whole lot of different viral RNA that's being made, but it's partial. You don't want to have a subgenomic transcript, for example, making it into the final viral particle. It would be defective. So full-length viral RNA contains a packaging signal, which is recognized by the M protein, so that other viral RNA, which many forms are produced, does not accidentally end up in the assembled viral particle. All right, so so far we've discussed, if you look at the genome, we kind of have a vague understanding of what these polyproteins do. So the non-structural proteins, they're involved in primarily replication, cleavage, replication, sequestering, and transport to an extent of the virus. Spike protein is fusion and cell entry. We talked about EM and N, but there's all this other stuff here, just an alphabet soup of 3A, 3B, 6, 7A, 8B. So these are so-called viral factors. So the question is, what do they do? And at the moment, the question is what components of the virus during the lytic cycle provided by the virus and what components provided by the cell machinery. So I mean life cycle, because virus generally doesn't necessarily immediately kill the cell. So for example, all the protein synthesis is utilizing host cell machinery, because you need ribosomes for that. And vesicle formation, transport, Golgi, the plasmic reticulum, all of this is standard part of the cell machinery. So, the virus only does a little bit. I mean, it only has about 20 proteins to play with. So it has to use, hundreds of cell proteins. And in fact, if you want to look at this detail, just Google for, just search Google Scholar or PubMed for, viral host protein interaction network. It'll show you exactly what we think is happening and which human genes or proteins are being used by the virus, but a lot. So basically, what virus does, it encodes some very specific things like its own proteases and its own RNA replication machinery, because it needs to do all these weird things like subgenomic transcription that cannot be done with cellular machinery. But the rest of the genome is where a lot of the sort of lack of our knowledge presently is, because you can do experiments to say, I'm just going to synthesize a viral vector that has everything except for these factors. Is it still able to replicate? And the answer is yes, it is still able to replicate in vitro, but it doesn't infect cells. It doesn't work in vivo. So what do they do? What do these quote unquote factors do? So factors, in other words, to say it does something, but we're not entirely sure what it does. The other interesting observation is whereas non-structural proteins are typically conserved, relatively conserved between coronaviruses because they more or less do the same thing. And a lot of them are even conserved outside coronaviruses, and you can find homology to other proteins. The ORFs, these factors, are highly variable between coronavirus species and seem to be changing dramatically between hosts. So for example, in SARS-CoV-2 and SARS-CoV-1, ORF3, which is right here, this little red blob, in SARS-CoV-2, it has a truncation relative to SARS-CoV-2. Basically, a truncation just means that a part of this protein isn't made. There's a stop codon in the middle, and the protein is shorter. So that is a dramatic change. For example, if you took the spike protein and just randomly lopped off 25% of it, it's just not going to work. All of it is used for something. It's a complex and fairly conserved life cycle, so it can't afford to do that. So in this particular case, you seem to be able to do that. So like they're the fudge factors. That's something that allows the viruses to, a degree of genetic flexibility. Genetic variability in ORF8, for example, is also very high, right? So, and if you look at the variation in what we see in SARS-CoV-2, there's a lot of mutagenesis, or not mutagenesis, but a lot of substitutions that occur at ORF8. If you're looking for answers about what these things do, I don't think we have these answers. We're going to look for them, or we're going to discover them, but we don't have a whole lot of answers yet. And we'll come back and look at specific proteins, later throughout the course, on and off, when it becomes appropriate. But generally speaking, a lot of these factors basically deal with host regulation and response. So, one of the things, for example, that the virus has to do is once it gets into a host cell, it wants to basically monopolize all of its cellular machinery to do nothing but make more virus. One of the mechanisms through which this happens is that the viruses are able to suppress or just cut up messenger RNA of host genes. That way, because ribosomes in the cells are not making host proteins, they'll be more available to make viral proteins. And as a byproduct, if you mess with gene synthesis, or so biosynthesis of gene to protein, you might also interfere with innate immune responses, because they obviously involve gene expression and protein synthesis. So for example, NSP1 is involved with mRNA repression, mRNA cleavage, and ORF7A has been shown to reduce overall protein synthesis. So that goes back to the previous question about, how do you hijack, what it is that you use in the cell. And, protein replication, protein synthesis is definitely something the viruses use. You also have immune antagonism that comes into play. You want to reduce immune response. For example, again, we're going to talk about various aspects of the immune response in separate lectures, so I'm just going to give you a very high-level overview. So for example, MERS downregulates interferon beta response, 3 times more, sorry, typo, than HCOV 229E. And that is important because you can show in experimental measurements the injection of interferon beta dramatically reduces, and I mean dramatically 10,000-fold reduces SARS-CoV-1 RNA copies in cell culture. And in vivo, interferon A reduced viral titer in macaques by an approximately same factor. So clearly, RNA copies you want to maintain. So virus needs to suppress such responses. On the other hand, some of these viral factors might be involved with stimulating pro-inflammatory host response. The other term for this, which you might have come across, is cytokine storm, which is usually associated with incredibly strong inflammation and poor outcomes. And this is usually, or very rarely, seen with endemic coronaviruses. So cytokine storm often is a marker for severe disease. So, one of the things that coronaviruses do is they downregulate, sorry, the number of typos in this lecture is greater than my threshold. I should include exonuclease into, my typing, because I have too many errors. So downregulation of pattern recognition receptors, so PRRs. So PRRs are host cellular proteins that are involved in innate immune response. And they will recognize sort of fairly non-specifically specific types of molecules. So for example, this RIG1 preferentially recognizes short double-stranded RNA. And MDA5 preferentially recognizes long double-stranded RNA, which is formed as an immediate during RNA copying. So you can see how this might interfere with the viral life cycle, because if you have human host responses coming in and degrading double-stranded RNA during one of the replication cycles, that prevents the virus from completing its cycle. So these need to be suppressed, because it does have to exist double-stranded RNA at some point. And several SARS-CoV proteins antagonize the host innate immune response. So ORF3b, ORF6, NSP1, NM, and NSP3. And as a final cartoon, here's an example, again, of our very partial understanding of where different viral proteins might interfere with the immune response. So all these red boxes here, you might recognize some of the abbreviations from the slides today. So these are all viral proteins. So N, protease, NSP1, ORF3b, nuclear protein, NSP1, ORF6, NSP1. And these are cellular response proteins. So NF-kappa B, interferon, signaling, JAK-STAT, which is a very common pathway to interfere with. So there's, for example, what I talked about in the previous slide, where you take viral double-stranded RNA and it's degraded by the immune response. So a cartoon like this, well, rather than being incomplete, shows two things. One, it shows the complexity of the interaction between any virus, really, but coronavirus in particular, and the host system, because it's always, the host cell has a lot of defense mechanisms which need to be overcome in order for the virus to successfully, even before adaptive immune response comes up, specifically to this virus. This is all non-specific, right? It doesn't care which virus it is. It's just generic response to infection or the presence of a particular type of molecule in the cell. And each of these interactions could potentially be viewed as a therapeutic target. Because if you were able to demonstrate that, if you're able to introduce an agent that prevents, some of these steps from successfully completing, then you don't necessarily destroy the virus, but you allow the cell to destroy the virus for you. The last thing, the last thing is a pro-inflammatory response. So SARS-CoV protein specifically, remember, it's in contrast to more benign endemic coronaviruses, have been implicated in modulating pro-inflammatory immune responses, so the cytokine storms that are detected in infected patients. And this usually has to do with interactions with protein kinases, so MAPK pathways that normally function to relay environmental stress to host cellular stress responses and trigger appropriate responses, for example, cell proliferation, differentiation, development, inflammatory responses, or in an extreme case, cell death or apoptosis. So three major APK pathways in mammals are all targeted during SARS-CoV infection. These are the three: the extracellular signal-regulated kinase, P38-MAPK, and the stress-activated protein kinase. And the reason for this, a correlation, for example, is that SARS-CoV-2 infected individuals show high levels of cytokine interleukin-1b, which is associated with an inflammatory form of programmed cell death. And ORF3a protein, for example, induces necrotic cell death. And if you have enough of those, you basically have a feedback cycle of inflammation response. This is where the inflammatory response is worse than the actual disease, because it could become a life-threatening.